All right. Hey, how are y'all? I am sorry I'm not here, but I hope you all enjoyed your break. I will hopefully see y'all on Wednesday, but today what I want you to do is go ahead and in your journals, you should have before Christmas finished all of these osmosis and diffusion vocabulary words. And so what I want you to do is on your first available page, on the right side. So make sure you're on the right side. So even if you have to flip the page and then start on the right, that'll kind of get us set up for correct on the rest of the section. So you're going to go ahead and set your paper up like this, and let's just checking to see. So we're going to talk about movement of stuff across a cell membrane. So we will use different colors in here. You don't have to use everything just like me, but let me zoom in on this just a little bit. So set up your paper, and where there's a blank, you don't have to draw a blank. You can just fill it in as I go if you want to. But the cell part that we're going to be talking about with excuse me, osmosis and diffusion is the cell membrane. So far we've talked about genetics, and we're dealing with the nucleus and the DNA inside of there. Biochemistry, which is just, you know, all of the small parts, and then we talked about protein synthesis, and the ribosome is the cell part for that. So the cell part for the movement of stuff in and out of a cell is the cell membrane. And something important that I really want you to pay attention to, it's not going to matter as much to you right now as it will as we learn about all these different types of cells, is that cell membranes surround all cells. We're going to talk about some cells that have outside of the cell membrane. They might have a capsid, or they might have a cell wall or something like that, but there will be a cell membrane there. This is the kind of the thing that allows stuff to come in and out of cells. It's for maintaining homeostasis. So this can also be called, and I'm having to kind of move around because I got this camera in the way. This is also known as the plasma membrane. So you might see it called the plasma membrane. So I just want you to have that there, so it's not, you know, you'll know what that is. So all cell membranes are made of three basic parts. Now sometimes they have, you know, different things to do what they need to do as the cell, but they have three basic parts. They are all made of a lipid bilayer. And bi means two, so it's two layers of lipids, proteins, and carbohydrate chains. And we're going to look at all those and how they kind of what they're doing. But the main function of the cell membrane is to maintain homeostasis. That is the function of the cell membrane. And what that means is that homeostasis means that everything is in balance of a living thing. So whatever it needs, it has, or it's allowed to get it. Whatever it needs to get rid of, it can move in and out. So what that means is that it keeps the cell in balance with whatever that cell needs to function properly. So it does this by bringing in what the cell needs and getting rid of what it doesn't need. Okay, so I do want to color code this in a certain way. We're going to look at the three main parts, the lipid bilayer, the proteins, and the carbohydrates. And I want to show you real quick, if you hold your hand like this and make a peace sign, that's basically a lipid. You have a hydrophilic head, and we're going to draw this here in a minute, but it's a hydrophilic head, and hydro means water and philic means loving, and so it's a water-loving part, and then two hydrophobic tails. And hydrophobic means, phobic is fearing, so it's water-fearing. And the reason you have to have a bilayer or two layers of these is because if you have two these two tails that will not touch water, and you throw it into water, then they're going to try to be away. They repel water, like an oil or something like that. So when you have another lipid and you throw these into water, they're going to automatically line up like this, and their tails are going to be on the inside. And you can see this down here. The tails are on the inside away from the water, and the hydrophilic or water-loving heads line the inside and the outside of the cell. So they're the ones that are going to be exposed to water. The tails are safe from water, so that's why it's a bilayer, is so that these tails are away from the water. So we're going to look at the lipid first. And I'm going to use like a yellowish color, but I'm afraid if I use yellow you can't see it, so I'm kind of using a peach. You're welcome to use yellow. But the first thing is a lipid. And we're going to draw it. It has this hydrophilic head and two hydrophobic tails. And so this 2 layers is a bilayer. And I'll just explain to you why it has a bilayer. Forms all cell membranes. Okay, so these 2 layers form all cell membranes because these will not be exposed to water. Now, sometimes we have things in our bodies, vesicles, and we're going to talk about vesicles later, that don't have water on the inside, and so it can be a single layer of these, and these can be on the inside because it's like some other type of liquid that's not water-based. So that can happen, but for cell membranes, they're all a bilayer because of these hydrophobic tails. So let's look at that. Hydrophilic. Head is right here, and I really should have drawn that, I guess, closer. And the 2 hydrophobic, or I'm sorry, I got ahead of myself. Hydrophilic means water loving, and 2 hydrophobic tails, which mean water fearing. So they're fearing, or they repel because of their chemistry. They repel water. Okay, so also membranes are made up of a bilayer of that. They're touching each other and they completely encircle the cell. The next thing I'm going to use, well, I guess before, I keep getting ahead of myself. Go down here to this picture and take the color that you used for your lipid. And I prefer yellow on this part just because, I don't know, I think of lipids being yellow. And just kind of color that in a little bit, just kind of squiggly on the tails, and just color those in with that same color. I'll give you just a second to do that. Okay, so the next part, I'm going to use green. And these are proteins. Well, we've talked about protein and how shape determines function. Shape always determines function on protein. And the proteins act as a transport option, basically, to go through the cell. So the protein is embedded. And there are different ones that do different stuff, but they're embedded in and through the lipid bilayer. And these are for transport of solute or molecules. So something that's dissolved in the cell, or something that's dissolved outside the cell that needs to come into the cell, or water or oxygen or whatever, glucose, stuff that needs to get into the cell, can do that through protein channels. And so down here, I'm going to take the same color, green, and these are the proteins right here. And just color those in. These are, terrible drawings because I did it. But I kind of tried to give you 2 different examples. One of them, and we'll get into this later when we talk about diffusion, but this is just kind of like a tunnel. You drive through a tunnel, through a mountain, it's just open. If you can fit, you can go kind of thing. That's what this is. This one changes shape when the right, if you'll remember, we talked about enzymes and how they have like active sites that make them change shapes and make things happen. This is similar to that. This is just a protein like channel or pump. It does change shape when the right molecule comes in. It'll change shape and then it'll open up on this side and then let it come through. So these are two different examples of protein. And so up here I'm just going to kind of draw a little tubey thing, just kind of like that, just a little tube, and they're shaped differently and whatever, but you'll see them in the picture. So I didn't finish that. So they're channels or tunnels. You know, just something that allows molecules to move, sometimes freely, sometimes not freely. And then the last one is our carbohydrate chains. And carbohydrate chains are, you'll see them located, and I'm just going to kind of do a little, because these are just the carbs coming off of here, on the outside of cell membranes, and they are for cellular communication. Carbohydrate chains are on the outside because we'll see how cells can communicate with each other. You know, pass on information from one cell to the next. They can also let each other know that, hey, you're touching another cell, so don't keep replicating, that kind of stuff. So just whatever they need to communicate to each other. So we're going to look at two new words real quick before we draw our carbohydrate chains. Extracellular, actually I'm going to use a different color to kind of make it stand out just a little bit. Extracellular space. And if you've ever heard of extraterrestrial, those are, you know, aliens or whatever outside of our Earth. And extra means outside. So this is outside the cell. Okay, so this is the space outside the cell. And intracellular space. Intra means inside, so this is inside the cell. Space inside the cell. So, you know, this would be your cytoplasm down here, and this would just be the space outside the cell. If it's skin cell or something, you would really be butted up against another cell, and they would be joined together so that you have a continual, you know, it's constant. If you're talking about blood or something, then these are all like floating, and so it just depends on the cell as to what's going on. But carbohydrate chains are outside of the cell so that they can communicate with other cells, and they're shaped different ways. So just draw a couple of different little carbohydrate things like that. Just a couple of different shapes, but they all need to be outside the cell like that. So that's just kind of the makeup of the cell membrane, and you know, just a very generic version of what it could look like. So if you flip the page, you have this little cell membrane picture, and what I want you to do is draw this concentration gradient. Okay, just real quick, I want you to pause me and glue this down. I gave you this picture, and if you don't have it, it's over there on the yellow shelf. And then draw this. Make sure that you have the big pink dots that I have here. Make sure that you have more on this side than you do on that side, noticeably more. And then I do want, it doesn't matter what color you use there, but I would like for you to use red or pink or something that we haven't really been using. And then do use blue for the tiny dots, because that's showing you water. So we're going to set that up and go ahead and pause me, and then we'll go from there. Okay. So what we have to do is talk about concentration gradient when we understand Uh-oh. Please. Okay, sorry if you heard me go, uh-oh, and please, and all that kind of stuff. It's because my computer went off, and I didn't want to lose this whole thing. Okay, so concentration gradient is just what makes things move. If you have ever had, spray perfume, then where that perfume comes out is extremely high concentration. It's very, very strong right there. But if somebody's standing across the room, they don't immediately smell it because there is no perfume molecules over there yet. But molecules bounce off of each other. Atoms are all the time. They're in, motion, especially if they're a gas. And so as they bounce off each other, they will try to spread themselves out into an equal concentration. And so concentration gradient just means it's like a pressure that's created because of a high concentration in one place and then a low concentration somewhere else. So it's kind of a little bit funky, but you've dealt with it before, even though you don't, may not realize it. But it's just where molecules are trying to move to make themselves either equal across a membrane, or if they're going up a concentration gradient, that means they're going to where they're more concentrated. So they're trying to build up some type of concentration for some reason. So we'll look at a couple of those. So molecules may need to get into or out of a cell through the cell membrane, but not all molecules are allowed to go through, which we're pretty excited about, because you do not want all viruses allowed in. You do not want a whole lot of something your cell doesn't need to be able to take up space in there. Remember, a cell is an individual living thing, and so if it's taking up all the space, then there's no room for the stuff that the cell does need. So cells don't just let everything move in and out freely because they are selectively permeable. Okay, selectively permeable is basically, your border patrol. They don't just let everybody in, and which is very important for the cell. So it keeps the cell able to function like it should. Their ability to move freely through the cell membrane can depend on a couple of things. So if something is allowed to move freely, such as water can move freely because it needs to move in and out for different reasons inside the cell. But things can move freely depending on their size, the cell's need for it, for whatever it is. If the cell needs it, it may let it in now. Later on, if the cell doesn't need it, it may not let it in. And the concentration gradient. Okay, and so think of it. Concentration is not how many particles of solute are there. Solute is what is dissolved in water. The concentration of something is how high the percentage of solute to water is. So if I had a cup of Kool-Aid in one glass, and then I had another glass that had Kool-Aid, but one cup, they had the same amount of water, but one cup had 3 times the amount of Kool-Aid powder in it, that is a much higher concentration. Even though I have the same amount of liquid, the one that has 3 times the amount of powder as the other one has a much higher concentration. So if you're given a cell membrane and the Kool-Aid is allowed to pass, then it will move until they're both the same concentration, until they have the same amount of Kool-Aid powder. So I hope that makes a little bit of sense. But what you get with the gradient is the difference in concentration of a certain molecule on one side of the membrane compared to the other. Okay? And so sometimes the cells don't want there to be an uneven concentration. Sometimes that, whatever it is, needs to be in equal concentration. Sometimes it does need an unequal concentration. So let's look at what we call this stuff. So in your little picture that you set up, this side over here, this is solute. Okay, this is solute, and then these are water. And if you'll remember from biochemistry, water is the universal solvent, which means it dissolves the solute because it's polar. So it's kind of negative and kind of positive. So it will dissolve all other polar things and ionic stuff. So things that are truly charged. And so solute, whatever this is dissolved over here, but there's a lot more solute concentration over here than there is over there. So what we call this, and I'm going to use red, this side would be hypertonic. Tonic is kind of referring to the concentration of something. Hyper, when you say somebody is hyper, that means they have a lot of energy. So you're talking about more or a higher concentration of solute over here. Now this side has a lesser concentration of solute, and so we call this hypotonic. Tonic is concentration. Hypo means less. So if you've ever heard of like hypothermia, that means thermia is talking about temperature. Hypo is low, so that's like where you're freezing. So hypotonic is less concentration. And you can see, if you can't easily see from the picture you drew when you pause me, then you need to make sure this side has a lot more concentration of solute. So you need to fix that, okay? So right down here, just say that the left side, which is this one right here, the left side is hypertonic compared to the right side, which is hypotonic, because there is. So this is hypertonic because there is more solute on the left than on the right, okay? Now, when we get to this, you're going to have to be very careful that you know which one we're discussing. So you have to pay attention. Am I talking about the stuff in the cell or out of the cell, or on the left side or the right side, or whatever? Because I could say the same thing right here. The right side is hypotonic compared to the left, which is hypertonic. So I can kind of switch it around. So you have to be careful and pay attention. So this creates what we call just like an osmotic pressure, and it's just a pressure where the solute will either move. So if this solute can move, then it will, because it doesn't want to be crowded. It wants to be even. If it can move, it will, and that when the solute moves is called diffusion. Or if the water is the only thing that can move, it will, which water would come this way, create more liquid, which dilutes this side, less liquid, which makes this side stronger. We'll look at all that later. That's called osmosis. Because life systems try to be in homeostasis, which the balance, that balance we'll talk about later, it's called isotonic, so it's where everything is equal. All right, so what I want you to do now is watch and do the worksheet for the cell membrane crash course.